So we're going to finish glycolysis today, if this will stay up. So we introduced it from the perspective of just thinking about like energy, right? And the idea that within metabolism, there's catabolic pathways and there's anabolic pathways. Catabolic pathways are those that oxidize carbons with the goal of producing energy so that we can use that in our bodies, right? And the anabolic pathways are those that will synthesize biomolecules, right? And this oftentimes involves reducing carbon, right? So building up biomolecules. They're opposing pathways. And I bring that up because today, once we finish glycolysis, we're going to start thinking about how to reverse glycolysis. So we're going to see our first anabolic pathway. Okay, we talked about all the intermediates of glycolysis, all of the enzymes that are important, and kind of talked about some nomenclature tips. Hopefully you all have that down and you're fairly comfortable with it. And then on Thursday, we started with the reactions, and we're going to finish that up today and then move into gluconeogenesis. So hopefully you still have your activity from last week. That we're going to finish that up today. Okay, but before we go into some of the new stuff, I thought let's just take a few moments to review where we're at and what we've seen so far. So an important thing to remember is that in glycolysis, there are three irreversible steps. And in metabolism, we define irreversible as something that's highly exergonic, right? Things that are close to equilibrium are reversible. So in glycolysis, there are three irreversible steps. They're all coupled to ATP in some way, right? Three of the four steps that involve ATP consumption or production are irreversible. So the first step is our committed step. That's where we convert glucose to glucose 6-phosphate. This is highly exergonic, very spontaneous, irreversible. This is important because it commits glucose to glycolysis. It also traps it in the cell, okay? The second irreversible step is the second phosphorylation, putting the second phosphate onto fructose. Okay, we produce fructose bisphosphate, and then we have several reactions at equilibrium, right? All the way to the very last step, which correlates to converting phosphoenol pyruvate into pyruvate, right? And in the process, transferring a phosphate from PEP onto ADP to make ATP. The only step that involves ATP that's not irreversible is converting BPG to 3PG, right? This one is reversible, okay? It's kind of easy to not remember that, but just remember that that one is reversible under the concentration that the cell maintains. Okay, so we have these three irreversible steps. We talked about the first 5 reactions, right? Ultimately going from glucose all the way to almost to 1,3-BPG. Okay, we'll quickly review these reactions, talk about the important features, and then we'll move on to the new ones. And for the most part, the new ones are quite straightforward. Okay, remember with hexokinase, the idea is really straightforward. We have a nucleophilic hydroxyl group on carbon 6. It becomes activated by deprotonation in the active site of the enzyme. It can then serve as a nucleophile and reach out and remove the phosphate from ATP. A very straightforward mechanism, about as simple as we can get in biochemistry. Okay, the important thing to remember about this enzyme is how kinases work. Remember, with kinases, the idea here is that ATP hydrolysis is just as likely to happen as phosphate transfer, right? So we talked about using this hydroxyl group as a nucleophile, but water can just as easily be a nucleophile, attack the gamma phosphorus, and that results in a more spontaneous reaction, right? Simply hydrolysis of ATP is more spontaneous than transfer of the phosphate. So kinetically, there's no difference. Thermodynamically, it's more favorable to hydrolyze ATP. So the idea here is this enzyme has to have a way of preventing the more spontaneous reaction while still allowing the less spontaneous one to happen. And we talked about how that boils down to this induced fit mechanism where there's different structures of the enzyme, right? So here we're looking at the open configuration before glucose binds. We see that it forms this nice cleft, this groove where glucose binds, and glucose binding triggers a structural change that closes that cleft. And once the cleft is closed, then ATP can bind, and that's when the reaction can happen. So ATP simply cannot get to the active site. It cannot bind to the active site until glucose is already there. And that's how we prevent ATP hydrolysis, because the only possible nucleophile at this point is glucose. It's that same idea that is persistent when we're thinking about other kinases. Always remember, these kinases are always function through an induced fit mechanism. They always have some sort of cleft closing which prevents ATP binding before the cleft closes, and that prevents ATP hydrolysis. Step 2 was isomerization of glucose 6-phosphate to fructose 6-phosphate. Okay? We saw this mechanism way back at the beginning of class when we talked about isomerases. And we see this very same mechanism later on when we convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. The idea here is that we need to be in the linear form, right? So it's easiest to visualize this in the linear form. In fact, it's not possible to do the mechanism with not being in the linear form. And the idea is simply that we have a general base in the active site that will deprotonate carbon 2 if we start with the aldehyde. In this case, we are. This moves the electrons up to the bond between C1 and C2, and we end up, so then we have a general acid up here, which will then allow us to pick up a proton, yielding this ene-diol intermediate, right? So between carbon 1 and carbon 2, we have a double bond. On carbon 1, we have an OH. On carbon 2, we have an OH. And then it can now reverse. So this is now a general acid, and over here we, this is general base now, and over here we have a general acid. So this process can now reverse, and we can deprotonate carbon 2, electrons flow over, we pick up a proton, and that then effectively shifts the carbonyl from C1 to C2. Okay? The trick here is recognizing that this has to start with deprotonation of a carbon, not deprotonation of an oxygen. If it started with deprotonation of oxygen, you would end up breaking the CC bond, and that's can't happen. Okay, so remember, this is initiated by deprotonation of either carbon 1 or carbon 2, depending upon which direction you're going. No matter which way you're going, you always go through this ene-diol intermediate. Come on. Oh, I hate you. Okay. So now we come to phosphofructokinase. This is where we put on our second phosphate with ATP being the source. This mechanism is identical to hexokinase, to step 1. And the important thing to recognize here is the enzyme functions in the same way, this induced fit mechanism. Our substrate, fructose 6-phosphate, has to bind first. It binds, it induces a cleft closing that allows ATP to bind. This is how we prevent ATP hydrolysis. We're going to see today there's other really important features of this enzyme that we're going to talk about that become very critical for glycolysis and gluconeogenesis. But for now, just understanding how it works, it's the same as hexokinase. Okay, step 4 is aldolase. This is also one of these reactions that we saw in the first weeks of class. The idea here is this is where we split our 6-carbon chain into 2 functionally equivalent 3-carbon chains. So we break the bond between C3 and C4. Okay, where the isomerization is initiated by deprotonation of a carbon, this one is initiated by deprotonation of the alcohol group on carbon 4. So we're going to see it's initiated by deprotonation of this alcohol group. Okay, the electrons flow over. That gives us our carbonyl on carbon 4, which ultimately is going to become the aldehyde on glyceraldehyde. The electrons then flow up to the bond between C2 and C3, and then will ultimately move up to an electron sink. Okay, we talked about that in bacteria, this is how it works. This oxyanion, this enolate, is stabilized by a magnesium that's positioned there. But in eukaryotes, in most organisms, it's actually not stabilized. Instead, we go through a shift base, right? So there's a lysine in the active site that reacts with carbon 2, and we make this shift base. By forming this shift base, we prevent the production of that high energy oxyanion. And since we're able to prevent that, then the reaction can be a lot faster. right? Otherwise, the mechanism is identical. We have a general base in the active site, deprotonases hydroxyl group, makes the double bond between the carbon and the oxygen. Electrons between C3 and C4 move up to make a double bond between C2 and C3. Electrons then move over to the Schiff-based nitrogen, our electron sink. This has created really our two products. We've now made glyceraldehyde 3-phosphate, right? Here's GAP, one of the products that move forward in glycolysis. And we've also made dihydroxyacetone phosphate, but it's still coupled, it's still covalently bound to the nitrogen, right? To that side chain lysine. So we need to remove it. The way this works is, remember with these Schiff bases, with these electron sinks, when you put electrons onto them, immediately reverse it and make the chemistry happen that we need to. So in the next step, the electrons flow down from the electron sink, the double bond breaks, we pick up the proton that we took off of that oxygen, and we now have a tetrahedral C3. C2 is linked up with the Schiff base, but remember, this can easily be hydrolyzed off. So then water comes in, it replaces the double bond between C2 and the nitrogen, releasing our ketone product, dihydroxyacetone phosphate. So this is DHAP. Y'all, I don't know what's going on. Okay. Right, so we looked at this structure where in bacteria, the Schiff base doesn't form. Instead, there's a metal ion that's positioned to stabilize that intermediate where we have a lot of electron density. Okay, this brought us to our second isomerase. This is TPI, triose phosphate isomerase, also called TIM. Okay, and we talked about structurally how this enzyme creates a structural motif called the TIM barrel. Okay, it's alpha helices outside flanking this beta barrel on the inside. And on the very center of this kind of wheel is the active site. And there are two important amino acids. There's a general acid, there's a general base. As we can see here, the general base in this case is glutamic acid. The general acid is a histidine. The mechanism is identical. Initiated by base deprotonation of, in this case, carbon 3. So base deprotonation of carbon 3. Electrons move over here to make that double bond. We now have the double bond here. We still have our OH. So this is reversed now, and we pick up, right, this moves over, get out of there. We pick up that proton, blah, blah, we reverse it, right? Exactly the same as the phosphoglucoisomerase. Initiated by base deprotonation of a carbon, we get this ene diol intermediate, and then it reverses, and we move the position of the carbonyl from C2 to C1. We switch from a ketone to an aldehyde. Identical mechanism as the glucose isomerase. We also mentioned that there's this cool feature of this enzyme that prevents the release of an otherwise toxic intermediate. So this ene diol intermediate can easily hydrolyze, removing the phosphate, giving us this molecule methylglycol, which is glyoxyl, sorry, which is really toxic. So there's this loop on this enzyme that flops over, places this isoleucine over the active site and prevents the release of the intermediate. Okay, so kind of like we talked about with flavin, with FAD, how the enzyme binds really tightly and prevents the release of potentially toxic things. The same thing goes here. During the reaction, there's a loop that flops over and prevents the release of that potentially toxic intermediate. That now brings us to where we kind of left off on Thursday, GAPDH. This is an absolutely critical, essential enzyme. Every organism on the planet requires this. If you take this gene away, if you mutate it so it doesn't function, things die. This is the only enzyme in glycolysis that's absolutely required for everything. Removing it is lethal. There's no way around it. We have to be able to interconvert GAP and 1,3-BPG. Okay, so this is where we really break between phase 1 and phase 2. This is at this point, We've produced 2 equivalents of GAP, and we now want to create our first high energy intermediate. And at the end of class on Thursday, I gave you the opportunity to start thinking about how this reaction happens, okay? And hopefully on your activity, you've kind of started moving through that. The general idea here is that this reaction happens in 2 steps. The first step is the oxidation, and the second step is the phosphorylation, right? It's an oxidative phosphorylation. The first step, we oxidize our aldehyde to a carboxylic acid. That generates 2 electrons that's given to NAD+. So we produce the carboxylic acid, we produce NADH, the reduced cofactor. This reaction is highly exergonic. We have a free energy change of negative 50 kilojoules per mole. So it's really exergonic. And this is in spite of the fact that it's actually not spontaneous to reduce NAD+, right? Reduction of NAD+ is not spontaneous. So the oxidation of this carbon produces massive amounts of energy, right? And that's why catabolism works, is because oxidizing carbon produces tons of energy. And that's the energy that ultimately we get to use to make ATP. So the idea that this reaction is really, really exergonic and the next reaction is really endergonic is a big problem, okay? So if we think about this in the context of a reaction progress diagram, right? So we've seen this before. On the y-axis, we have the free energy. On the x-axis, we have the progress of the reaction as we proceed from reactants through our transition states, through our intermediate, and then ultimately to the products. So here, this would represent our reactants where we have GAP, we have phosphate, we have oxidized NAD+. This is the first oxidation step of it. And remember, this has a very exergonic delta G, which means that this intermediate is a very, very low energy. So the next step, going from our oxidized product, the carboxylic acid, up to our product, which phosphorylates that carboxylic acid, is highly endergonic, but also there is this massive, massive, massive activation energy. And that's a problem, because what does a giant activation energy mean? It has nothing to do with spontaneity. Remember, the difference between reactants and products has to do with thermo. What does activation energy have to do with? Kinetics, yeah. Right, so these diagrams show 2 things. They show the thermodynamics, the overall spontaneity, the difference in energy between reactants and the intermediate, or the intermediate and the product. But the activation energies correspond to our rate constants. Remember, K equals A e to the minus EA over RT. Y'all remember this? Those of you who've been to the PK, I'm sure as hell better remember this, right? So the bigger this activation energy is, the smaller the K is. Everybody's like, yeah, sure, I remember. So the bigger the activation energy, the more negative the exponent, and the smaller the rate constant. Small rate constants means really slow reactions. So this becomes a huge problem. Even though this reaction overall is spontaneous, even though step 1 releases enough energy to drive step 2, in theory, the second step is so slow that it may not happen. In addition, the second step has such an endergonic energy that perhaps we can't do it. So there has to be some way of preventing this, we call it a kinetic or thermodynamic trap, right? It's a potential energy well where once you're in it, you can't get out, right? Imagine like literally falling down a well. It's hard to climb out. And that's the idea here, is you can't get out once you're in. So the way that this enzyme has evolved to prevent this is instead of going through a carboxylic acid, we go through this thioester, okay? Which is functionally identical to a carboxylic acid, except the oxygen is replaced with the sulfur of a cysteine side chain. So it still involves the oxidation of that carbon, but we don't have the carboxylic acid. Instead, we're trapped in this thioester. And the beauty of that is that this bond is a high energy bond. So we've trapped energy now, right? We have trapped a lot of the energy of oxidation of that carbon. It's now trapped in that thioester bond. So instead of going all the way down into this potential well, instead, the energy of this thioester intermediate is roughly the same as our products. And that dramatically decreases this activation energy. And that's an activation energy we can overcome. So this is an important theme, right? We consistently are talking about how it's important to be able to trap energy. This is one way where we can trap energy in covalent bonds. This is how we translate the energy of oxidation of that carbon to something that's useful, right? If the carbon oxidized all the way to the carboxylic acid and we didn't have this intermediate, we would just release all of this energy and it would be gone forever. Instead, we trap it in this bond, which we can then use in the future, okay? And in fact, we use some of it in the next step, because in the next step, we bring in inorganic phosphate. It reacts with that carbon and releases the cysteine, okay? This is important. We're going to see this again. We see this in the TCA cycle, right? The formation of these thioesters is a great way of trapping energy in a useful manner. All right, so let's think about how this reaction works. Okay, it really does follow along really, really well with what we just talked about. Okay, so here's the overall mechanism. Let's zoom in. Okay, so in the first part, we have an empty active site. Note here that the thiol is deprotonated, and that's because the chemical environment of the active site switched the pKa of the cysteine so that it's more acidic, so it's simply deprotonated. Right, and if you don't know how that happens, we can talk about that some other time. So we have an active site that contains the oxidized NAD. We have NAD+. We have a deprotonated cysteine, so it's genuinely ready for this oxidation reaction to happen. Our substrate, GAP, comes in, and the first thing that happens is that the sulfur of this cysteine attacks the carbonyl. Okay, exactly what we'd expect if water or hydroxide is our reactant. But instead, the nucleophile is this thiolate. Okay, we get this intermediate, our tetrahedral intermediate we've seen before, and when it collapses, instead of breaking the CS bond to the CC bond, we release hydride. And that hydride is the source for the reduction of NAD+. Okay, we now have this intermediate where we have the reduced NADH. The carbon has been oxidized, so this carbon has now been oxidized, but it's trapped covalently linked to the enzyme through this thioester bond. And that bond is holding energy for us. That bond makes it reactive. So in the next step, we can bring in phosphate. We can swap out our reduced cofactor for our oxidized cofactor. And this phosphate can then readily react with that electrophilic carbon, right? So the carbon here is our electrophile. We can go through the same type of carbonyl chemistry we talked about before. Nucleophilic attack of the phosphorus, of the phosphate. We would create an intermediate, and this time when the intermediate collapses, we release the sulfur. So we've broken that covalent bond, and our product, 1,3-BPG, can now exit, making a new active site that has the deprotonated cysteine, or not a new active site, but regenerating the active site that has oxidized NADH, or oxidized NAD+, and a reactive cysteine. We're now ready for another substrate. So the chemistry is not surprising, I don't think. Once you know that this thioester forms, then the chemistry is pretty straightforward. Instead of hydroxyl or water being the initial nucleophile, we have this sulfur from cysteine being the initial nucleophile. The tetrahedral intermediate collapses, releases hydride, and that hydride reduces NAD+. Phosphate then becomes a nucleophile, attacks the carbonyl, releases the sulfur, and that creates our products. Any questions about that? Okay. So if we look in the active site of GAPDH, it has exactly what we expect, right? So what we're looking at here in purple, this is our NADH. If you were to look closely, we would see that this is a Rossmann fold that recognizes this dinucleotide, right? So it binds to our dinucleotide. We have the nicotinamide site. The dot that I have there, that's the site of the reduction. And note here that it's positioned towards the gap, towards the binding pocket where our gap normally would bind. We have our cysteine is positioned perfectly, and we have this histidine that's positioned to help orient our substrate. So it has everything we expect. All right, next step is another kinase. So this is phosphoglycerate kinase. Remember, when we create a high energy intermediate, we use the high energy intermediate. So we create a reactive phosphate, we're going to use that reactive phosphate. So GAP has a reactive phosphate. It has enough phosphoryl transfer potential to phosphorylate ADP and make ATP. That's exactly what happens. This enzyme functions in the same way you would expect a kinase to function. Okay, there's no surprise. You have a binding cleft, same type of induced fit mechanism, substrates bind, and we get phosphate transfer. But this time, phosphate transfer from GAP onto ADP to make ATP. Okay, just to reemphasize that we truly do have the same type of binding cleft. We can see here our binding cleft, we see our nucleotide binding site, and we see our substrate binding site. It's GAP here, not 1,3-BPG, otherwise it would react. Okay, we have now created 3-phosphoglycerate. The next step, remember, is to convert that to 2-phosphoglycerate, so we can then dehydrate 2-phosphoglycerate to get our second high energy intermediate, PEP. Okay, this is a fairly straightforward mutation, isomerization. Okay, but it proceeds through an interesting mechanism, right? So it proceeds in a way where we end up with this intermediate that has 2 phosphates on carbon 2 and carbon 3. So if you go back to hemoglobin, like way back to like we talked like with Dr. Hurlbert, you talked about hemoglobin and how there's this high altitude adaptation. Do y'all remember this molecule 2,3-BPG? This is the molecule that binds to hemoglobin and allows it to bind more like oxygen more tightly. This is the molecule that does it. It gets produced as a byproduct of glycolysis. Okay, so the idea here is in the active site you have a phosphorylated histidine, and that phosphate is the source of the second phosphate in our 2,3-BPG intermediate. Okay, it's a fairly straightforward mechanism. Go ahead and take a few moments to try to work through it by yourself.